All right, ladies and gentlemen, this is going to be the next video on active and passive transport. So let's get started on this next video. So there's two types of transport that we need to be thinking about. We have passive transport, and then we're also going to have active transport. Now, passive transport is a movement of ions or other atomic or molecular substances across the cell membrane without any energy input. Remember, the ions are traveling with the concentration gradient. They naturally want to go through and spread out, so therefore that requires no energy for the cell to move those ions across the membrane. Now, the next is active transport. An active transport, the cell must expend energy to pump a solute across the concentration gradient. So this is going to require work. Remember, we're moving against what the molecules or the ions naturally want to do. So therefore, the cell has to use ATP and use energy to move those ions from one side to the other. So when we're thinking of the three types of active transport, we have membrane pumps, which we just looked at. We also have exocytosis and endocytosis, and this is going to be for bulk transport of things across the cell. So what is the point of active transport? Well, active transport is going to use a molecule called adenosine triphosphate. And adenosine triphosphate is going to be the energy carrier for active transport. And active transport enables a cell to maintain internal concentrations of small solutes that differ from the concentration of the environment. So it essentially lets you pump solute across or against the concentration gradient where the ions wouldn't normally flow. So let's go through and look at a sodium-potassium pump to better understand this. So in a sodium-potassium pump, what we're trying to do is maintain concentration gradients that naturally do not want to occur on the exterior and interior of the cell. And the exterior here is the blue and the cytoplasm is the interior. So what we can see here is a sodium-potassium pump. On the exterior of the cell, we have a high concentration of sodium ions and a low concentration of potassium ions. And then over here, we have a low concentration of sodium ions and a high concentration of potassium ions. Now, cytoplasmic sodium is going to bind to the sodium-potassium pump. When it's open, when it's kind of tilted open on the cytoplasmic side, the affinity or the tendency for the sodium to bind to it is really, really probable or really, really high. Now, the concentration normally wants to flow this way, but as the cell, we're trying to pump them against the concentration gradient. Remember, the sodium wants to flow down, the potassium wants to flow up, but we're trying to build sodium on the top and potassium on the bottom, so we're going against the concentration gradient. Because we're going against the concentration gradient, we're going to have to put in ATP. It's going to require energy for us to go through and pump sodium up and potassium down. So once the sodium binds, it's stimulated. The protein is stimulated by phosphorylation of ATP, and it results in a conformational change which pumps the sodium up into the exterior of the cell. So when we see this, the phosphorylation, or when one of the phosphates broke off the ATP, we get a change reducing the affinity for Na+. So now that it's opened this way, it doesn't want to bind with the sodium anymore. It wants to bind with the potassium. So what we see is the potassium now that the sodium's on the exterior of the cell, the potassium's going to bind with the protein. There's a higher affinity, and we're going to get potassium to go through into this pump. And as a result, it's going to go through another conformational change, which is going to result in potassium being pumped down into the cytoplasm. Remember, against the concentration gradient. So here, the new shape is a high affinity for K+, which binds on the extracellular side. Now, this is really, really important for neural function. Your neurons function based on these sodium-potassium pumps. And all of the electrical activity is dependent upon polarization of sodium and potassium inside and outside of the cell. We also have to think about chlorine too, but you will learn that in an AP biology class if you go through and take that. Essentially, when a neuron fires, it's depolarizing. These ions go through and flood into the neuron, and we build up the polarization. We build up that concentration gradient to get the neuron to fire. So different concentrations of sodium and potassium create a gradient and electrical impulse, allowing you to think and allowing you to move. So if we're comparing this, well, right now we're looking at passive transport, not active transport. Remember, passive transport, we're flowing from concentration of high amounts of ions to a concentration of low. If we're thinking about passive transport, we have diffusion. So diffusion is going to happen. It's hydrophobic molecules and very small uncharged polar molecules that diffuse through the lipid bilayer. And again, if we're looking at this type of passive transport, we can use carrier proteins, facilitated transport, also known as facilitated diffusion, where we have many hydrophilic substances that diffuse through the membrane with the assistance of transport proteins. So we either have channel proteins, which are on the left here, or carrier proteins on the right. Now, active transport, well, this is different. Remember, we are pumping against the concentration gradient. An active transport is when transport proteins act as pumps, moving substances across the membrane against their concentration gradient. This requires energy for this to occur. So, bulk transport. This is another type of active transport. We're going to look at a concept map just so we can understand this. But we have two types of bulk transport. We have exocytosis, and then we're also going to have endocytosis. Now, exocytosis is the bulk transport of material out of the cell via fusion of vesicles with the plasma membrane. Endocytosis is transport of material into the cell by forming vesicles from the plasma. So let's go through and look at these in a little more detail. So when we were talking about this endosymbiont theory, well, this was a perfect example of endocytosis, or phagocytosis, in which the endosymbiont, or the mitochondria, which was a bacteria, was engulfed and consumed by a larger cell. All right, so if we're looking at exocytosis, we have a vesicle which is going to be formed by phospholipid bilayer, and let's say we want to remove whatever these pink particles are. Now, what this is going to do is it's going to fuse with the cell membrane. Remember, the cell membrane is all made out of the same thing, phospholipid bilayer. It fuses with the bilayer, and then as a result, what it creates is a little pocket that ejects all of the material outside of the cell. That's exocytosis. Now, in endocytosis, we're moving stuff into the cell. So again, we have these purple particles here, cell membrane, vesicle. Remember, they're both made out of the phospholipid bilayer. So it goes through and it pinches off a small section in this small circle. And by pinching off a small amount of that phospholipid bilayer, what we get is a sphere that can go through and contain the particles that we need, capture them in a vesicle, and move them to somewhere else, maybe the mitochondrion, or maybe a lysosome where they can get broken down, maybe used for parts or used for energy. Now, if we're thinking about endocytosis a little more specific, we have two different types. We have phagocytosis, and phagocytosis is when the cell engulfs a particle, which we kind of generally refer to as cellular eating. So the cell is engulfing a large solid particle that's going through and being contained in a vesicle. Now, the next type is going to be penocytosis. And penocytosis is when the cell engulfs large amounts of fluid. So we're going to refer to this as cellular drinking. Now, in penocytosis, we're going through and capturing fluid and putting it in the vesicle, whereas in phagocytosis, we're consuming a large particle and containing it in a vesicle. So if we're trying to understand all of this, let's go through and look at a concept map to kind of better understand where we are. So when we're thinking about this, we have passive transport. And passive transport does not require energy. So requires no energy. Now, there are different types of passive transport. We have diffusion, which is just going to be across the lipid bilayer. And then we also have facilitated diffusion, or facilitated transport, or passive transport. And in this one, we're using proteins to get those molecules across. But remember, they're going with the concentration gradient. Now, active transport, we're going against the concentration gradient, so therefore this is going to require energy. Now, we have multiple types. We have active transport via membrane pumps, which is just going to be the movement of small materials through channel proteins via ATP. And then we're also going to have endo and exocytosis, which is bulk transport of materials, which again are going to require ATP, and they're going to form vesicles, and they're going to use the lipid bilayer to form those vesicles. Now, everything over here in the yellow is going to transport small amounts of material. So ions are very small molecules, whereas everything over here in the pink is transporting large, or it's bulk transport, the transport of large materials across into the cell or across the membrane. Now, everything over here does not require energy and moves with the concentration gradient, whereas everything over here requires energy and moves against the concentration gradient. So if we're looking at this, passive transport goes with the concentration gradient, therefore requiring no ATP. Active transport requires energy because it's moving against the concentration gradient. So did you guys learn? Well, did you learn a couple of things? Did you learn active versus passive transport? Did you guys learn which types require energy? And then did you learn how cells move small and bulk material across the membrane? This is going to be the end of the video. I will see you guys in class tomorrow.